Hi students, welcome to SPSVERSITY. In the previous session, we discussed about Thomas Hunt Morgan experiment, the linkage and recombination. So now we are discussing about the concept of mutation. What is meant by mutation? So mutation is a sudden change that is in the phenotypic. And this sudden change is due to change in DNA or gene sequence. So the organism which undergoes mutation, it is called mutant. Okay. So change in DNA sequence or gene will lead to mutation. What changes will cause mutation? What are the changes? Like, for example, if there is a change in chromosome, there is in the structure of chromosome, if there is loss or gain of a segment of DNA, so these are called chromosomal aberrations. That is, change in gene chromosome is called chromosomal aberrations. If change occurs in gene, that is due to change in a single base pair of DNA, it is called gene mutation or point mutation. The best example for this is sickle cell anemia. So here, due to change in deletion or insertion of base pairs of DNA, it is called the frame shift mutations. It is called frame shift mutations. One base pair will change the next sequence of DNA. So that's why it is called frame shift mutation. So this is about a brief of mutations, and the substances which cause mutations, they are called mutagens. So in this there are physical and chemical mutagens. So the substances which will change, which will cause mutations, they are called physical, or that is, there will be physical or chemical mutagens. Okay. So here, this is the concept of mutations. In this, the gene mutations, the chromosomal aberrations, and frame shift mutations. After this, we are going to discuss about pedigree analysis. What is meant by pedigree? So the pedigree is the study of genetic traits. So the traits which come from several generations in a family. You can take whatever trait, like color, height, like we can take any of the traits in a family and their inheritance pattern. So this study of inheritance of traits is called pedigree analysis. It is done in a specific family. What is the purpose of this? The purpose of this is it helps in genetic counseling to avoid genetic disorders. That is, the couples who are going to marry, they are given genetic counseling based on their pedigree analysis. So what type of the offspring, the children are born, what type of genetic disorders possibilities are there. So that is called genetic counseling, which is done for the couple which are going to marry. So this pedigree analysis helps to trace, that is, to find out the inheritance pattern of a particular type. So in this pedigree analysis, many symbols are used to represent different individuals. Like for example, if you take male, male is represented by a square. Female is represented by a circle. Sex unspecified, that means you are not able to find it out. This is the symbol. If these symbols are filled with the color, like whatever it is, like black or red, whatever it is, it shows affected individuals. Affected means they are having that specific trait. If you want to write the mating, that is marriage between male and female, a line is drawn between pair and a circle. If you want to represent, that is, if you want to represent a close marriage, that is marriage between close relatives, two lines are drawn. If one line, simple mating. If two lines, it is consanguineous mating. That means mating between relatives, very close. And when you come to here, this change, which that is the children who are born to this couple, they are written like this, in the order of left to right. That is, here if you take this example, the first child is a baby girl, the second child is a baby boy. Okay. Suppose if this is infected, if this boy is infected, it is filled with the red color. The female is not filled means it is not affected. Okay. And here also there is a representation for pins. If you draw lines between that is the hops, the children, one line between identical things. If we don't draw any lines, that is not identical things. So like this, we use symbols in pedigree analysis for representation of a pedigree chart of a family. So whenever you take a family, so you can see the picture here very carefully. So in this picture, if you take the next, that is, these are parents. This is parental generation. So these parents, if you study very carefully this pedigree chart, these parents has 3 sons and 2 daughters. That is, the first son, the second son, third is a girl, daughter, fourth is daughter, and fifth is, that is a son. So total they are having 5 children. In these 5 children, 1 son and 1 daughter is affected. It is filled with color. That means they are affected. That means it shows the particular trait. And remaining are not affected. These are not affected. Remaining 3. So the first daughter, that is the third child, that is the third one, married, and she has 1 boy, 2 girls. In that, 1 is affected. So it is filled with this one. So like this, you can write the pedigree chart of any family until you know the history of the family, like taking the parental generation, the next generation, and the next generation. This is generation 1, and this is generation 2. Okay. So this is generation 1, this is generation 2. Okay. In generation 1, in generation 2, suppose if you take generation 1, there are 5 children. Okay. So this 5 children, this is, you can mention child number 1, 2, 3, 4, 5. Here also you can mention child number 1, 2, 3. In generation 1, the second and fourth child are affected. In generation 2, first child is affected, remaining or normal. Affected means it shows the specific trait which you are doing a pedigree analysis. So this is one example of an pedigree chart. A pedigree chart looks like this. So you can write the data, whatever the data we have, with the available data, we can write the pedigree chart. Okay. So this pedigree chart is useful not only to trait to specific trait, it is also useful to study disorders. What disorders? Genetic disorders, chromosomal disorders. So when you come to genetic disorders specifically, these are of two categories. One is Mendelian disorder and another one is chromosomal disorder. So what is meant by Mendelian disorders? So Mendelian disorders are due to change, that is mutation in single gene. Okay. And this disorder, that is, it obeys the principle of Mendelian inheritance. That means it is transmitted from one generation to other generation. Chromosomal disorders means where there is a change in chromosome structure or chromosome number. So what examples will come under Mendelian disorders? That is hemophilia, color blindness, cystic fibrosis, sickle cell anemia, phenylketonuria, thalassemia. So these all will come under, that is, Mendelian disorders. That is, genetic disorders are classified into Mendelian disorder and chromosomal disorders. This is classified into chromosomal. So first we'll discuss about Mendelian disorder. So what is the reason for causing of this? That is due to change in a single gene locus. Okay. So this gene which is present, it may be on a sex chromosome or it may be on an autosome. Okay. So whatever the gene which is controlling the specific trait, it may be present on sex chromosome or it may be present on autosome. Okay. So if it is present on sex chromosome, that is, if it is present on sex chromosome, it is called sex link. Okay. If it is present on autosome, it is called autosomal. So when you are writing sex linked means the gene is concerned is present on sex chromosome. Autosomal means it is present on the autosomes. That means in sex linked mostly they will be present on X as well as on very few will be present on Y. Autosomes means the remaining 22 pairs of chromosomes. Okay. And here if you see the diseases one by one, if you see this, in this sex linked and autosomal, again there are two subtypes. They may be recessive or dominant. Here what is meant by recessive, what is dominant? Recessive means when they are present in, when both the genes are recessive alleles. When the both alleles are of recessive, then the disease will come. If any one is dominant allele is present, then the disease will come. This is called dominant mode. So here you can see phenylketonuria is an autosomal recessive disorder. It will come when both the alleles are recessive alleles, small p, small p. Cystic fibrosis, when both are small c. Phenylketonuria, small p, small p. Sickle cell anemia, when both are having S, that is small s, small s. small a, small e. When both the alleles are recessive, then the disease will come. Dominant means when both the allele, when any one of the allele is a dominant allele will get. So first we'll discuss about sickle cell anemia. What is meant by sickle cell anemia? So sickle cell anemia is one of group disorder, that is sickle cell diseases. Okay. So what happens in sickle cell anemia? That is, it is related to red blood cells, where there are no enough healthy blood cells. When there is no sufficient quantity of red blood cells, it affects oxygen carry throughout the body. Okay. If oxygen is not properly carried throughout the body, it will affect the metabolism of the body. Okay. So due to this, due to this sickle cell anemia, the enough red blood cells will not be there, where the oxygen will be affected. Okay. And next thing is, normally red blood cells are round, which can move easily through blood vessels. But with sickle cell anemia, the red blood cells cannot move easily. Due to the change in the shape, what happens is they cannot move easily through the blood vessels, so that they get clogged in the blood vessels. So here what happens is, due to shortage of red blood cells, it leads to sickle cell anemia, and blocked blood flowing cause sickle cell crisis. Due to block of blood flowing, it causes pain. It is called sickle cell crisis. So we'll see what is the reason, what is the cause for this. Okay. So in this sickle cell anemia, it is related to red blood cells. In red blood cells, again there is a pigment called hemoglobin. So this cause, the reason for cause this is, there is a change in the gene of hemoglobin. Here it is given C. In normal, that is normal hemoglobin, if you see the normal hemoglobin, HbA. In this, if you take the 6th amino acid, that is glutamic acid, normally, but here it is replaced by valine. What is the reason for this? The reason for this is the replacement of adenine, that is A means adenine. Due to change in one single base pair, that is adenine is replaced with thymine. So that what happens, obviously the amino acid also, instead of glutamic acid, it comes to valine. That means in hemoglobin protein chain, in the chain of protein, a change is caused in the sequence of amino acid, that is in the place of glutamic acid, valine has come. What is the reason for this is due to change in one single base pair, that is adenine is replaced by thymine. Okay. Due to this, what happens, valine has come. Okay. So here this, due to this sickle cell anemia, infections, pain will come, even it leads to fatigue. Fatigue means that easily get tired. Like it includes medication, blood transfusions, and really bone marrow transplant. So now we studied, that is, what is the cause for sickle cell anemia. So this is the picture. This is picture of normal red blood cells. Normal or B. So this is the picture of sickle cell. The red blood cells or of the shape is changed to sickle cell. So that's why it is called sickle cell anemia. As the shape change, they cannot carry oxygen. and they get clogged in the blood vessels. See the difference. Due to just change in one single base pair, it has affected the shape of RBC. As the shape of RBC is affected, the functioning of RBC also affected. That is, oxygen carrying is affected. And next, that is, due to this sickle cell shape of RBC, the RBC gets clogged, the blood gets clogged in the blood vessels, which leads to sickle cell crisis. So this is how we can see the shape, change in the shape of RBC in sickle cell anemia. So here when it comes to the study of sickle cell anemia inheritance. In inheritance, as it is in sickle cell anemia is an autosomal recessive disorder. That means the gene which is responsible for sickle, that is the shape of RBC, it is present on autosome. How to represent that genotype? The genotype is normal individual. Normal individual is represented by HBE. The genotype for normal individual, that is without sickle cell anemia, is HBE. Carrier means he doesn't suffer with disorder. But the next generation, the possibilities are there, that is HBE, HBS. That is affected with individual, that is affected with sickle cell anemia. The genotype is HBS. So when both are HBS, then they will get sickle cell anemia. So in this picture, if you take, the male is carrier, one sickle cell gene carrier. That means the genotype of this is, the male is HBE, HBS. Even the female is carrier, that is HBA, HBS. When both got married, so here you can see one individual, no sickle cell, that means it is not affected. One sickle cell carrier means the genotype will be HBA, HBS. It is not affected means HBA, HBA. Carrier here also, the female one, that is one is carrier, that is HBA, HBS. Okay. And he is suffering with sickle cell anemia, that is HBS, HBS. That means when a male and female, when they are carriers, they got 4 children. In this, one is not affected, 2 are carriers, and 1 is affected. That means if you write the, that is if you write the with genotypes and with crosses like this, if you write, so HBA, HBS, that is if you write the gametes of these, it will be HBA, HBS. So here we will get 2 types of gametes. Here also HBA, HBS. We get 2 types of. When we cross these 2, here it will be HBA, HBA, normal. HBA, HBS, carrier. HBA, HBS, carrier. And the final one is HBS, suffer. So this is how we will do the crossing in case of sickle cell anemia. Suppose here if you write this, the genotype, that is if you write the pedigree chart of this, if you take the particular one, this one, You can write here the pedigree chart. This is male. This is female. Both got married. So it is both are carriers. Generally carriers are represented by, that is generally half. In most of the cases they are not represented. Any carriers will be represented. Okay. So they got married. They had 4 children. So in these 4 children, if you see, so they had 4 children. Okay. So in this 4 children, if you see this one, he is suffering with sickle cell. That he is not suffering with. Square without. A carrier. She is a carrier female. Okay. Observed. Okay. So he is, that is carrier male. Pair. Suffering with sickle cell anemia. The box is completely filled with the color. Okay. So like this we can write the simple pedigree chart so that we can understand how to write the pedigree charts of a particular disorder. So this is about the pedigree chart of sickle cell anemia. And next we are going to discuss about the next disorder, that is phenylketonuria. So this phenylketonuria is also an autosomal recessive disorder where the genes which are related to phenylketonuria, they are present on autosomes. So what happens in phenylketonuria? We'll see what is the reason for phenylketonuria. So it is an inborn error of metabolism. So what will happen is, due to this phenylketonuria, the phenylalanine metabolism is decreased. So phenylalanine is an amino acid, essential amino acid which is required for our body. Okay. So here there is a classical phenylketonuria is an autosomal recessive disorder caused by mutation in both the alleles. So what will happen is, and the genes are present on chromosome 12. So here what happens is, in this disorder, due to mutation of both the alleles, a phenylalanine hydroxylase enzyme is not synthesized. What this phenylalanine hydroxylase will do is, it will convert phenylalanine into another amino acid, that is tyrosine. This is general conversion takes place in our amino acid metabolism. But due to lack of this enzyme, this conversion doesn't take place. When this conversion doesn't take place, what happens is, this phenylalanine converted into phenylpyruvic acid and gets accumulated in the body. Okay. So this phenylalanine hydroxylase will convert amino acid phenylalanine to tyrosine. So tyrosine is again another essential amino acid. So when it is not treated, this phenylalanine is converted into phenylalanine pyruvic acid, which will harm the brain cells. It leads to intellectual disabilities, seizures, behavioral problems, and mental disorders. So what is the treatment for this? That is a strict diet with limited protein. And if you see this, that normal, this is okay. So here in normal, phenylalanine is converted to tyrosine. It is normal metabolic process which takes place in our body. But due to this phenylketonuria, this enzyme is not secreted. So what will happen is tyrosine will be decreased and phenylalanine will be increased. Okay. When phenylalanine increases, what will happen? You can see. Okay, echisma, hypopigmentation, less pigment, seizures, limb spasticity, mousy odor, mental retardation. So these are symptoms of the phenylalanine. And while you are writing, as it is a phenylketonuria is an autosomal recessive disorder, you can write, that is, the genotypes. While you are doing a crossing, you can write the genotypes. Like you can mention, that is normal. So normal means, that is capital P, capital P. Carrier, capital P, small p. Affected, phenylketonuria, which is affected with small p, small p. So these are the genotypes which you write for doing crossings. So it is also an autosomal recessive disorder. And next we are going to discuss about hemophilia. The genes which are related to hemophilia, that is, they are present on X chromosome. That is called sex-linked recessive disorder. What will happen in hemophilia? The individual who is suffering with hemophilia, the blood will bleed continuously. Even for a simple small injury also the blood will bleed. And the gene for hemophilia is present on X chromosome. It is not present on Y. Okay. So what will happen? That is, what is the reason for this? That is, if you take the coagulation of blood, the process of coagulation of blood, so many proteins are involved in this. A cascade of proteins, that is, a sequence of proteins are involved. In this, one protein that is involved is affected. Due to this, the coagulation doesn't take place. And this, as it is present on the gene which is related to this, as it is present on this X chromosome, generally in this such type of disease where the gene which is present on X chromosome, mostly males will get the disorder. Very rare cases female will get the disorder. We'll see the reason for this, where the gene which is related, it is present on X chromosome, it is not present on Y. It is not present on Y. So here you can see. So while we are writing the genotype, for example, father with hemophilia. So father with hemophilia means you can write like this, or you can mention like this, as it is autosomal recessive disorder X small h y. Even one X with small h, it will, the male will get the disorder. Unaffected female, that is, the genotype we can write is X capital H, X capital H. So what are the possible outcomes for this? So when you do crossing for this, the gametes for this male will be X small h. The gametes for this is X capital H. When you do crossing, one male, that is one female, that is XH, XH, she is carrier. whereas another male, that is XHY, normal. That means while you are writing the genotypes for hemophilia, you can write specifically for male. Normal male, what is the genotype? X capital H Y. Hemophilic male, what is the genotype? Hemophilic male means X small h y. There is no carrier in males. But when you come to females, normal, X capital H, X capital H. Carrier, X capital H, X small h. Sufferer, that is suffering with hemophilia, hemophilic female, X small h, X small. As it is recessive, both alleles should have small letters. Okay. And as these genes are present on the, that is X chromosome, as they are not present on Y chromosome, even in males, if one X chromosome, one X small h is present, they will get the disorder. But in females, it is not the case. Both should be with recessive alleles, then only they should get. So this is about the hemophilia disorder, which is a sex-linked recessive disorder. This is a sex-linked. So here you can see. Father unaffected by hemophilia. That means you can write the genotype for this is X capital H Y. Mother carrier of hemophilia, X capital H, X small h. Now do the crossing between. What gametes will get here? So here if you take the gametes will be, the male gametes will be, when you take the male gametes, you will get X capital H, Y. Female gametes, X capital H, X small. Do crossings. So here what will you get? Normal female. It is normal female. X capital H, X capital. Okay. Next, carrier female. X capital H, X. That is a daughter carrier, that is X capital H, X small h. Next, normal male, X capital H, Y. Next, hemophilic male, that is X capital H, Y. That means here if you take total out of 4 individuals, that is 25%, that is if you take females specifically, 2 are there. So 25% normal, that is 25% in females, 50% normal, 50% carrier. If you take males, that is if you take 50% normal, and if you take here 50% carriers. That means if you take the total entire 25, 25, 20. So this is how we'll see, we'll get the final outcome of this offsprings. Okay. So females who are suffering with disease, generally they are suffering from the disease means they will have homozygous condition. Homozygous means suffering with the disease, X small h, X small h. And in this case, the female becoming hemophilic is very rare. What is the reason is, in female, at least one, the father, suppose if you want to get a female, the outcome of female should be a hemophilic means, the mother should be carrier, at least carrier, and the father should be hemophilic. In such cases only you will get the hemophilic female. That means, in a cross where mother is carrier and where father is hemophilic, in such cases the chance of getting a 25%. And generally affected transmit the disease only to the son, not to the daughter. Daughter can receive the disease from both mother and father, but it is not in the case of son. Son can receive from mother only, no chance from father. That is why. Okay. So this is about hemophilia. You can do the analysis of hemophilia. Okay. Here it is given C, sex linked. If you see this cross very carefully, there is 25% normal, that is carrier female, 25% suffering males, female suffering, 25% normal, 25% suffering with male suffering. That is, the ratio is 1:1:1. That is, in females, one is carrier, one is hemophilic. Okay. And in males, one is normal, one is hemophilic. That means if you write very carefully here, X capital H, here it will be like this. Okay. So you can write like this. So this is the analysis of hemophilia. You can see here the pedigree chart. So this is affected one. That is, the male is affected. The male is affected means you can write the genotype. Female is unaffected. So they have 2 sons and 2 daughters. So one son is affected, X, H, Y. Female is affected, that is X, H, X, H. Another female also affected, X, H, X, H. So that means the mother is carrier. See here, the first child in the second generation, they have 2 children, one female is affected, X, H, X, H. Okay. And the second son, if you see the second one child in the first generation, he is not affected, he married a normal female, no female hemophilic here. But see the last girl, that is last child where she got married, one son has hemophilic and one daughter he is hemophilic. So this is how pedigree. And next we are going to discuss about color blindness. So what is meant by color blindness? So this is a X-linked, that is sex-linked recessive disorders where they cannot identify few colors. That is, if you take the human retina, it contains 2 kinds of light cells. One is rod cells and another is cone cells. Cone cells are active in daylight and responsible for color perception. Rod cells are active in low light. That is, you can take in the dark. So here, when you take the cones, they are meant for color perception. Red cones, which perceive red color, red cones, green, blue. In that red cone percentage will be 64, green cones 32%, blue cones that is 2%. So here there are different types of color blindness. One is monochromatism, where the condition will reach the complete color blindness, in which all the colors will appear green. But when you come to dichromatism, in which the partial blindness, where 2 colors are perceived, whether red, green or blue, yellow. And there is daltonism, where the color cannot identify red, green color. They cannot differentiate red and green. So they cannot differentiate red, green and yellow colors. And the protanopia, where in which the perception of red and confusion of red with green or bluish. Actually it will be red, but the perception will be either green or bluish. This is protanopia. Deuteropenia, where a form of color blindness characterized by insensitivity of green. They cannot identify the green. The green looks green. Okay. And while you are writing the color blindness, the genotypes for color blindness, we can write X capital C, X capital C. That is normal female. If we write X capital C, X small c, carrier female, same like hemophilia, but only letter change. X small c, X small c, that is color blind female. But when you come to males, it will be normal male XC capital Y, normal male. X small c Y, that is color blind male. So the same like you mentioned the in hemophilia, but here we'll write in the place of H, we'll write C, remaining all same. Here also the same formula applies, that is generally males are more affected than female. The reason is the gene related to this is present on X chromosome. And next, that is thalassemia, which is also related to the red blood cells. Okay. But it is not same for, that is the sickle cell anemia. So here you can see thalassemia is caused due to change in mutations of hemoglobin. So it is an autosomal recessive disorder. Okay. And this thalassemia is passed from parents to children. So here you can see normal or BC, which will be normal shape. Thalassemia or BC will be in different shapes. It's not like that is the disc shape, it is of different shape. What is the reason for this? So the reason for this is, if you take hemoglobin, in hemoglobin there are alpha beta chains. In alpha thalassemia, the gene related to alpha globin protein, that is alpha hemoglobin protein, are changed or mutated. Due to this mutation, what happens is the alpha globin protein chain is secreted less, and the genes HPA1, HPA2 are present on chromosome 60. So here in this, if you see this, it is called alpha thalassemia. Beta thalassemia means the genes which are related to beta globin chain are affected. These are genes are HPB, which are present on chromosome. That is, the synthesis of beta chain is impaired. Here when you take thalassemia and sickle cell anemia, thalassemia is an quantitative disorder, whereas sickle cell anemia is a qualitative disorder. Both are related to RBC, but there is a difference. So here you can see the clear structure of this. Normal red blood cells which has globin, heme, iron atom. So red blood cells will carry oxygen by binding to hemoglobin. Okay. Oxygen is carried through blood by blending with oxygen with hemoglobin, which forms oxyhemoglobin. Okay. What are the symptoms? Thalassemia can cause anemia, which leads to fatigue very easily. Okay. So it requires regular blood transfusions, and there is a cheilious therapy. There is treatment to remove excess of iron from your blood, and there is stem cell transport. Okay. And next one is, that is, you can take steps to fatigue. Fatigue means you can take steps to fatigue, that is which is choosing a healthy diet and doing exercise regularly and taking green leafy vegetables. Okay. That is the greeny leafy vegetables which should be avoided, which are rich in iron, so which are put not to eat for thalassemia patients. So till now what we discussed is the Mendelian disorders. In genetic disorders we discussed about Mendelian disorders. Now the next one we are going to discuss about is chromosomal disorders. So this chromosomal disorders means there is a change in chromosome number. Okay. And there is a change in structural of chromosome. So here you can see two types of chromosomal abnormalities. One is numerical abnormalities, another one is structural abnormalities. So here if you see, the most common type is numerical abnormality. Numerical means there is a change in chromosome number. Like you can see, there is aneuploidy, where it leads to loss or gain of one or several individual chromosomes from diploid cell. So that means if you take, for example, humans have 46 chromosomes. And in these 46 chromosomes, that is, one pair instead of two, they have 3. That is, it leads to trisomy. That means 47 chromosomes. Instead of 46, 47 will be. And this aneuploidy is results due to non-disjunction during meiosis, the process which takes place during meiosis. And next, polyploidy. Polyploidy means failure of cytokinesis, which leads to increase in whole set of chromosomes. That means 46 can become 92. The whole set of chromosomes becomes double. So here we will study about a few autosomal disorders. One is Down syndrome, which is named after the person John Langdon Down, who first described this. Okay. And this Down syndrome is due to one extra chromosome. That is, in 21st pair, instead of 2, there are 3. That is, one extra chromosome 21 is there. Due to this extra 21 chromosome, one extra, you can see here, this 21st pair, one extra is there. Instead of 2, there are 3. Remaining all are having 2. That means here the total number of chromosomes will be 47. That means if you take autosomes, the autosomes count will be 45 and the sex chromosomes will be 2. So 45 plus 2. Okay. So 45 plus 2. Okay. So here you can see the symptoms. That is, the growth failure, mental retardation, okay, flat black of head, abnormal hair, special skin, intestinal blockage, hernia, abnormal pelvis, big toes, enlarged colon, congenital heart disease, wrinkled tongue. These all are symptoms of Down syndrome. So syndrome means it is not just one symptom, a group of symptoms. Okay. What treatment, that is help, requires a medical diagnosis, lab test and imaging. And in India, it is more than 1 million cases per year. And next is Klinefelter's syndrome. So in this Klinefelter syndrome, it generally occurs in males. In males, there will be, that is, if you take the male's chromosome number, there will be 44 autosomes plus 2 sex chromosomes. There will be total 46. That is XY. But here, there will be one extra X chromosome, that is XXY condition. That means 44 plus 3. That means one extra X chromosome. Instead of one extra X chromosome, there are 2 X chromosomes and 1 Y chromosome. It leads to infertility and poor functioning of testicles, that is testes. Okay. So it is not inherited, but it rather occurs due to genetic error. Okay. So generally it occurs in males. They may have less testosterone, reduced muscle, body hair, facial hair reduced. Okay. And these males will produce very little or sperm. So treatment includes testosterone replacement and fertility treatment. So here you can see there are 2 X chromosomes and 1 Y chromosome, remaining all our autosomes are normal. That means 1 extra X chromosome. Due to this, the total count will be 47. Here you can see the symptoms: baldness, poor beard growth, narrow shoulders, wide hips, small testicular size, long legs, pubic hair like female, breast development, tendency to grow fewer chest hairs. So these are the symptoms seen in KLINEFELTER'S syndrome. Even it leads to gynecomastia, that is male with female features, small testes, inability to produce sperms. Okay. So this is the about KLINEFELTER'S syndrome. So these are the symptoms: taller than average, longer legs, delayed puberty, less muscular bodies, less facial, small testicles, small penis, enlarged breast, gynecomastia, weak bones, low energy, shyness, socializing less, difficulty in socializing, problem in reading and writing, attention problems. And the next syndrome is Turner syndrome. That is, it generally occurs in females. So in females, if we take, that is, there will be 2 X chromosomes. By here 1 X chromosome is missing. That means here there will be X0. Due to lack of X chromosome, that is, we'll have total chromosome number will be 45. That means 44 autosomes, 1 sex chromosome. Due to this, the females will be short statured, delayed puberty, infertility, and learning disability. The treatment can be by hormone therapy and fertility treatment. It generally occurs in females. So here you can see the symptoms. That is, poor breast development, elbow deformity, rudimentary ovaries, that means non-functional, lack of proper menstruation, brown spots, small fingernails, shortened metaphorical in forearm, white space nipples, shield-shaped thorax, low hairline, short stature. These all are symptoms of the Turner syndrome, where there are 1 X chromosome is. The total count will be 45. So here you can see only 1 sex chromosome is present. So till now we discussed about the Mendelian disorders and the chromosomal disorders. Follow us on SPSVERSITY on social media. Thank you all. We'll meet again.